Excuse me, sir.
Oh, I'm sorry. Hi.
Can I ask you a question?
Okay, yeah.
What is apical dominance?
Dominance? Apical what? Hi. Welcome back to another edition of Plant Bio. Today our topic is going to be apical dominance. You may be wondering, what is apical dominance? Apical dominance occurs in most plants when there's a shoot apex right here. I'll label that. Inhibiting lateral buds right here. And what is the outcome of that? The outcome is that there'll be less branching when the shoot apex inhibits the lateral buds. And as you can see in this normal plant. But when there is no shoot apex, it cannot inhibit the lateral buds from growing, so there'll be more branching, as you can see right here. And as a result, most trees that you see outside form a triangular shape, and then we'll dive in more into that later.
Breaking news. Shoot apex of a real-life plant in Davis, California has been decapitated.
Let's cut live to the scene where we have a reporter and a witness ready to talk about this.
Breaking news! We are here at the murder scene with this witness. Please tell us what has happened.
There was this guy, and he had a knife, and he cut off the shoot apex of the plant.
It's okay.
What would happen to the plant?
We will tell you what will happen.
Hi everyone, welcome to episode 2 of Plant Biology. So now we're going to talk about the biological significance of apical dominance. So apical dominance, again, is where the shoot apex inhibits lateral bud growth. So this will lead to enhanced competitiveness of a plant in a way where apical dominance, it promotes vertical growth and the lack of horizontal growth. So as you can see, if these were two separate plants, plant number 1 will have enhanced light exposure, which will lead to enhanced photosynthesis, which will lead to enhanced fitness. So this is an adaptive property that apical dominance will cause. Hi everyone, welcome to episode 3 of Plant Biology. We're going to talk about another significance of apical dominance. So when a plant is decapitated, what happens? Does it die out? Does it just shrivel up and become organic matter for soil? No. What happens is that the plant is still able to complete their life cycle because of this factor, plant recovery. So apical dominance helps with plant recovery by ensuring that the plant has horizontal growth even when the shoot apex is decapitated. So this is a decapitated plant. There is still lateral bud growth so that the plant can still maintain and produce seeds that grow into new plants. So this helps the plant continue its life cycle even though the shoot apex is cut off. Hello, welcome to episode 6, I believe, of Plant Biology. Now we're going to go right into the mechanism of how apical dominance works. So the first hormone we want to introduce to you is auxin. Auxin plays a role in apical dominance in inhibiting bud outgrowth. So our first model here in this plant, where we have decapitated the plant by removing the shoot apex, which is the main synthesis location of auxin. So we've removed the shoot apex, which means the plant lacks endogenous auxin. There is bud outgrowth because auxin is not present. So auxin plays a role in inhibiting bud outgrowth, and when auxin is taken away, there is bud outgrowth here, as you can see. And in this second frame here, when we have decapitated this plant, again, we have lost the shoot apex, making it so that there is no endogenous auxin, but we have added auxin exogenously. We do not see bud outgrowth because auxin is an inhibitor of bud outgrowth. Another hormone we'd like to introduce that plays a role in apical dominance is cytokinin. First of all, cytokinin activates axillary buds, which we will show more in detail later. In addition, cytokinin is auxin's antagonist. Basically, these two hormones work in opposite of each other. This is shown in an experiment when auxin is added to a plant. The IPT gene, responsible for the biosynthesis of cytokinin, is repressed. So we'd like to introduce another hormone to you. This is strigolactone, a hormone involved in the apical dominance pathway. So first of all, strigolactone helps auxin relay information to other parts of the plant. We will explain this later to you. And second of all, it causes the transcription factor BRC1, which we will highlight in detail later on, that inhibits bud outgrowth to be expressed. So strigolactone promotes BRC1 transcription factor. Finally, strigolactone was found in plants that were mutant for strigolactone, which were found to basically be insensitive to auxin, because they were unable to relay the information from auxin to other parts of the plant.
So branch 1, also called BRC1, is a key transcriptional factor, and it's responsible for maintaining bud dormancy.
Alright, we're going to introduce another key player in apical dominance, which is sucrose. So the role of sucrose in apical dominance is that when we have the shoot apex, there'll be a high level of sucrose because the shoot apex has a high demand for sucrose. But if we decapitate the shoot apex, more of the sucrose would be devoted to the lateral bud, which would promote lateral bud outgrowth. So by increasing sucrose in the lateral buds, it would inhibit BRC1, which is the regulator responsible for maintenance of bud dormancy. So by inhibiting bud dormancy, it would result in bud release and outgrowth. So as there is less sugar going to the lateral buds right here, which are right there, there'll be a decrease in bud outgrowth. And also, with the shoot apex, there is a great amount of auxin, which would inhibit bud outgrowth. And over here, we have a decapitated stem. And without the shoot apex, the lateral buds will have more sucrose going into it because there is no shoot apex. So it'll be a greater amount of sugar, which would increase bud outgrowth, as you can see right here. And also, without the shoot apex, there's no auxin available, so it will not inhibit bud outgrowth.
In conclusion, sucrose demand, not auxin, is the initial regulator of apical dominance. And the main reason why is because sucrose can travel down the stem and around the plant as the main transport of sugar for the plant quicker than auxin is decreased in a decapitated bud. Now that we've gone through all the hormones one by one, let's put them all together in one pathway. So we have auxin to start out with as an inhibitor of bud outgrowth. But how does that actually happen? So first of all, auxin works to promote strigolactone and to inhibit cytokinin. We said before that auxin was an antagonist of cytokinin, so you see that here. Next off, strigolactone works as a communication between auxin and branch 1 to activate branch 1, and cytokinin inhibits branch 1. So in an intact plant, auxin will be active in the shoot apex, which will promote strigolactone and promote branch 1 to inhibit bud outgrowth. And in an intact plant, auxin will be active to inhibit cytokinin, which will stop its inhibition of BROC1. Furthermore, sucrose will inhibit branch 1, and let's look at which of these are positive or negative regulators. Auxin is a negative regulator of bud outgrowth, cytokinin is a positive regulator, strigolactone is a negative regulator, and sucrose is a positive regulator, and branch 1 is a negative regulator of bud outgrowth. So let's go through what happens in a decapitated plant now. So because there is no endogenous auxin at work because we got rid of the shoot apical meristem in a decapitated plant, there is no activation of strigolactone and there is no inhibition of cytokinin, which allows cytokinin to inhibit branch 1. In this case, branch 1 is not allowed to inhibit bud outgrowth. And furthermore, sucrose inhibits branch 1. And again, branch 1 is not allowed to inhibit bud outgrowth. And in this model, there will be bud outgrowth occurring because the negative regulator of bud outgrowth, which is branch 1, is not active.